Boa tarde a todos. Hoje a palestra da disciplina Seminários de Zoologia, do curso de pós-graduação em Zoologia do Museu Nacional e do curso de graduação em Ciências Biológicas, ambos da UFRJ, será sobre a imensa diversidade de formas larvais de um dos filos de invertebrados mais intrigantes, os nemertíneos, e seu significado evolutivo. Será ministrada por uma das expoentes no tema, que é a professora Svetlana Maslakova, do Instituto de Biologia e do Laboratório de Biologia e Instituto de Biologia da Universidade do Oregon, nos Estados Unidos. It's a great honor to have Dr. Svetlana Maslakova here with us to talk about the Nemertean larvae and their evolution. Thank you very much for accepting the invitation and being here with us. Dr. Svetlana received her degrees of Bachelor and Master with honor from Moscow State University in Russia and her PhD from the George Washington University in the United States in 2005. After this, she has had positions as research assistant at the Whitney Laboratory for Marine Bioscience at the University of Florida and postdoc researcher at Friday Harbor Laboratories of the University of Washington, both in the United States. Dr. Maslakova is currently Associate Professor at the Oregon Institute of Marine Biology and Department of Biology of the University of Oregon, where she has been since 2008. She runs the Maslakova Lab, teaches and supervises undergraduate, master's, and PhD students. She's also a guest editor for the Biological Bulletin. Dr. Maslakova has an outstanding career with studies on development, diversity, and evolution of Nemerteans and their larvae. Her main research interests are systematics, evolution, and development of marine invertebrates, in particular the very interesting phylum, the Nemerteans. So without further ado, here with you, Dr. Svetlana Maslakova.
Thank you, Joanna, for inviting me. It's a pleasure, and it's really fun too. Thank you, everyone, for being there for this talk. So I'll talk about my work on evolution of Nemertean larvae. Nemerteans, to give you a bit of an introduction, are a phylum of marine invertebrates. These are the perfect worms. They lack any kind of features whatsoever. They don't have segments, they don't have setae, they don't have any hard parts whatsoever, except some of them have little stylets, of which I will talk about a little bit later. As you can see on the slide, these worms can be really beautiful, colorful. Some of them have aposematic coloration, especially some of the tropical species. There's one group, this is one of the representatives of this unusual group of holopelagic Nemerteans. There's about 100 described species. These guys live in a water column at great depths, up to several thousand meters, and they're sort of semi-transparent. But most Nemerteans are benthic marine predators. And these guys use a unique structure that they have that is a synapomorphy of this phylum. It's this thing that sticks out from this pelagic Nemertean at its anterior end. It's called a proboscis. It's a long tubular organ that normally lies within a fluid-filled cavity, and it's everted rapidly, hydrostatically, and the Nemertean attacks its prey. There's about 1,300 described species worldwide, but my work on biodiversity and systematics over the past 20-some years shows that there is probably several times that number of undescribed species in the world. There's lots of cryptic speciation where things look alike but are distinct at the molecular level. And there's also just a lot of species that we didn't know were there. There's very few people who study these guys, so it's not a huge surprise. Anyways, this is a video of a live Nemertean worm from the Pacific coast of North America. It's a Cerebratulus herculeus. And looking at the way this worm moves, this you can grasp why the common name of these worms is ribbon worms. They sort of look like a ribbon as they swim along, undulate in their body. This is an infaunal species. Normally it lives in the sand, buries itself, and then at night they come up and crawl around on the surface, hunting polychaete worms and bivalves. That was the proboscis, just diverted briefly. That's the mouth here. This here is a video of how a Nemertean uses its proboscis. This is not my video. Oh, there it goes. That was the proboscis. That pink thing that just shot out from its anterior end. That was the proboscis. This is a video by a colleague of mine from Germany, Jörn van Doren, and it shows a Nemertean worm attacking a polychaete worm, a Nereid. And so what the Nemertean had just done was shoot out its proboscis, and the proboscis is covered with a sticky mucus, which contains neurotoxins, which paralyze the prey within a short period of time. And so this polychaete is now kind of in agony, slowly losing its ability to move. Here comes another one. The Nemertean is greedy. It shoots the proboscis again. And a short struggle ensues. The proboscis is sticking to the surface of the polychaete. And then once the prey is immobilized, the Nemertean swallows its prey whole, kind of like a python snake. So this feeding strategy of some Nemerteans is where the name pythons of the sea comes from, which was coined by a famous Russian zoologist some decades ago. It is because they can swallow very large prey. As you can see, the size of the polychaete here is comparable to the size of the Nemertean itself, and they swallow this thing whole and then can go for a long periods of time without feeding. And they lose their body mass as they starve, but they don't die. So they're really pretty easy to keep in the lab for that reason. Some Nemerteans have a little calcareous stylet mounted, so that's the stylet here, mounted on a proteinaceous basis of various shapes. And that ends up at the tip of the proboscis when the proboscis strikes, when it's everted from the body and strikes the prey. So presence of the stylets characterizes one group of Nemerteans, and that allows them to feed on prey that has hard exoskeleton. So for example, this green and yellow worm here is called Implectanema viride, and that's a predator of barnacles. They somehow manage to strike the barnacle between the hard plates and penetrate the soft cuticle and suck out the semi-liquefied contents. So the venom that they inject with the stylet, the venom produced by the proboscis epithelium and injected through the wound inflicted by the stylet, contains not only neurotoxins that paralyze the prey, but it also contains proteolytic enzymes which semi-digest the tissues, and then they become liquid, and the Nemertean simply sucks them out, kind of vacuums them out. On the right here, you can see an image of Carcinonemertes, which belongs to a family of Nemerteans that parasitize egg masses of decapod crustaceans, crabs, lobsters, and so on. And these worms are highly specialized to feed on the eggs of these crustaceans, and the eggs have a pretty hard eggshell, but they puncture it with their stylet and suck out the egg. And the worms need to feed on the eggs in order to reach sexual maturity. And then what they do is they lay their eggs among the egg mass of the crab, and their larvae hatch out and then swim off and feed in the plankton, as it turns out. We've recently discovered that. Nobody knew what they were doing in the plankton. People thought they were just settling right back on the crabs, but actually they spent probably several months in the plankton feeding on copypods. and then they find the crabs and settle down. They settle both on males and females, and then when the crabs mate and the male holds the female, the worms migrate to the female and wait for the eggs to be deposited to feed and then reach sexual maturity. So Nemerteans belong to this large clade on the bilaterian tree of life called Spiralia or Lophotrochozoa, depending on which you prefer. And it's one of the most diverse clades of bilaterians in terms of body plans. So this is a slide from a review paper on spiralians that was part of the virtual symposium on spiralian development some years ago. And it just showcases the diversity of body plans within this phylum. You can see a sipunculate here, a peanut worm, a bunch of different kinds of mollusks with their diverse body plans, polychaetes, phoronids, brachiopods, and Nemerteans, flatworms, echidurids, which are now part of annelids, bryozoans, very diverse body plans. But I mean, the adult body plans are just part of the story of the diversity of spiralians. The other part is the larval body plans. So this is a bunch of planktonic larvae of different kinds of spiralians. And some of them, it's pretty easy to tell who they belong to. For example, that's clearly a gastropod villager, and that's an annelid. You can see the city. And some of you may be familiar with some of these other larvae, and others, those of you who study terrestrial mammals, probably are not. So I'll just say that that's a horseshoe worm larva, a phoronid larva called the actinotrog. And this little triangle here is a bryozoan larva called Siphonautes. And this is a sipunculin larva, a peanut worm larva called Pelagosphere. That's a polychaete larva, Polygordius, that's another polychaete larva. And this is a Nemertean larva called the Pylidium. So I will introduce the Pylidium a little bit. And this is a video that my husband had composed from several videos, kind of superimposing them on each other. These are Pylidium larvae that swimming on the slide under the microscope. And this type of larva was discovered in 1847 by a German anatomist and physiologist and plankton aficionado, Johannes Müller. And he, during the summers, went to the North Sea and the Mediterranean and used the plankton net, which he made himself, to drag it through the water and collected all kinds of interesting things and the plankton and looked at them under the microscope. And he discovered and described many larval types of marine invertebrates. And so those of you who are not familiar with marine invertebrates or their development, I just wanted to point out that many marine invertebrates have biphasic life cycles. So the adults are benthic, and they have a planktonic life history stage called a larva, which can look very different from the adult because it is occupying a very different habitat and has different biology and a different role. And so Nemerteans, like many other marine invertebrates, have a biphasic life cycle, and most of them have one or another kind of larva. And the most charismatic type of larva in Nemerteans is this Pylidium larva. So when Johannes Müller first saw these things, he called them Pylidium gyrans. He didn't know what the hell it was. He had an educated guess that it is a larva of some kind of marine invertebrate, but he didn't know which. He named them Pylidium gyrans in reference to this gyrating motion. As you can see, they kind of rotate as they spin through the water. The scale bar here is, these things are about 500 microns in diameter. And Pylidium means, it's a word derived from the Greek word Pylidion, which means cap, a kind of brimless hat or cap that was worn by Greek soldiers. So it's a hat that spins through the water. And only some 20 years after Müller had discovered these little hats in the water, a Russian zoologist, Mechnikov, had described that it's a larva of a Nemertean. He, in fact, observed, if you can see, there's a little worm in the middle of that hat. And so that worm hatches out, and it is a Nemertean worm. So Meshnikov had discovered this connection and said, aha, Pylidium is the larva of a Nemertean. This is another video by George, my husband, showing the Pylidium larva through a compound microscope. These are alive and swimming, so you can just appreciate how transparent and beautiful these organisms are. It's really striking how you can see individual cells and even the nuclei within those cells. This is yet another view of a Pylidium larva. This one is preserved in formalin, and I stained it with a fluorescent label called phalloidin. Phalloidin is an interesting dye. It's derived from a toxin of a death-capped mushroom, Amanita phalloides, and it binds filamentous actin, of which, of course, there's plenty of in all cells, especially so in muscles, which are contractile. So actin is part of the contractile machinery in all cells. So these strands that you see here are muscles within the Pylidium larva. You can also see other things. For example, microvilli on the surface of many cells, they're packed with actin underneath the cell membrane. So this bright dot here up at the top is the apical organ, which is composed of many columnar cells with microvillar collar around the bundle of cilia. And these little dot, dot, dot along the main spanning these lobes and lappets and along the main ciliary band of the larva are the sensory cells within the ciliary band with which the Pylidium larva detects algal cells or other prey in the water, and it uses these muscles to capture the prey. So basically the Pylidium is like a feeding machine. And so this is a video of a Pylidium that's being held up by a suction pipette by its apical organ. And this is the juvenile worm developing inside the Pylidium larva. This pink blob here is the gut of the larva, and it's pink because it's been feeding on cryptophyte algae called rhodomonas, and they look kind of red. So it accumulates the pigment from the algal cells. And so the way the Pylidium feeds is you can see that it flicks its larval lappets and its lobes, and there's like a funnel there in between the lappets and lobes, and that's its mouth. And as it flicks its lobes and lappets, it traps individual algal cells. So these little dots you see flying through are the algal cells in the water. And the current here, a lappetward current from the apical organ toward the lappets, is created by the ciliary band of the Pylidium larva. It's not swimming because it's being held by the suction pipette, but it's creating a current, and you can see how the algal cells are streaming past the ciliary band, and when they are detected on the margins of these lappets and lobes, then the algal cells are captured. This is a slow motion video of this process, and you can watch how individual algal cells passing the rim of the lappets or lobes are then detected. There's a brief pause there, detection, and then a response by the larva that deforms the lappets or lobes, allowing it to gulp a parcel of water containing the cells. So it's a bit like a baleen whale. And we published a paper about it a few years ago, how the Pylidium larva feeds. And it really is remarkably like a baleen whale. Not only does it gulp a large parcel of water containing small particles, but then it sieves them out. It has kind of like baleen, only made of cilia, and barricades the exits while to keep the food inside its funnel and to expel the excess water. So here's a particle that's now trapped between the two lappets, and it's going to be consumed. So while the Pylidium swims around in the plankton, feeding on unicellular algae, it goes through this remarkable process in which it develops the juvenile worm inside its body through a series of initially isolated rudiments called the imaginal discs. This is about a 3-week-old pylidium of a local species here in the Northwest Pacific. It's called Maculaura alaskensis. And these lumps of tissue here are the imaginal discs. This is about 3-week-old. And so it has 3 pairs of discs. That's the gut. That's the apical organ with its long ciliary band, that ciliary tuft. That's the ciliary band that spans the lappets and the lobes. This is a slightly more advanced stage in which the discs, the cephalic discs, which will give rise to the head of the worm, the cerebral organ discs, which will give rise to a pair of unique sensory structures in the head of the worm, and the trunk discs, which make the trunk, are fusing with each other. And there is another pair on the other side, so that's a bilaterally symmetrical structure. So it forms kind of like a donut of tissue around the larval esophagus, which is this funnel here. And gradually this tissue grows over the stomach and over the esophagus, forming a complete worm. So this one's almost done with its development of the juvenile inside the pylidium larva, and there's like a little gap here left in the back. And what happens next is once the juvenile is complete, which usually takes several weeks or months in the plankton, depending on the amount of food they get and the temperature and so on, the species. Once the juvenile is complete, it's ready to undergo metamorphosis. And metamorphosis in the pylidium larva is catastrophic and rapid. So you capture one of these things in the plankton, so that's the larva here. It's about 500 micron tall. And the juvenile is inside, and it just ruptured the little amniotic membrane which surrounds it inside the pylidium larva. And now it's trying to rupture this very thin larval epidermis, and it just succeeded. And you can see its head sticking out and all the cilia beating on its surface of the juvenile worm. The problem with this is that the juvenile develops around the larval gut, incorporating the gut. And so there is a physical connection around the mouth, which is right about here, between the juvenile body and the larval body. And so in order to sever this connection, most juveniles begin to back out of the larval body while swallowing, slurping in the larval body. So the larval body is being pulled over the head of the juvenile and into the mouth of the juvenile. This is sped up compared to real time, about 2.5 times, just because it takes a little longer for these guys to do that sort of thing on the slide compared to when they're just in the water, in the bowl. So here is the juvenile worm that had just completed its metamorphosis and is now a benthic worm, a predator, and it just had its first meal of its own larval body. So this is a remarkable dramatic transformation. It's one of the most dramatic examples of so-called indirect development in the animal kingdom, where the larval body is so unlike the juvenile and adult body, and the transition between the two life history stages and the two body plans is rapid and catastrophic. So it takes literally a minute or a couple of minutes in this case. So I have always been interested in the evolution of larval development in this group, and I have wondered, how is it that this incredible diversity of larval body plans within the Nemerteans, how did they evolve? So you can see all of these different shapes here. Some of them look like hats, some of them look like little worms, so they're called direct developers, even though they have a larval stage, but that larva is very similar in its body plan to the adults. It doesn't have a catastrophic metamorphosis. So only one group within the Nemerteans has this development with the pylidium larva and catastrophic metamorphosis. And then there are these bullet-like or planula-like larvae that have a catastrophic metamorphosis, interestingly. And then there's some that look almost like trochophore larvae of annelids or mollusks. And then there's some that look like socks. And so I'll talk a little bit today about how this diversity evolved, or my insights through the years of research into how this diversity originated. If we map the developmental types of Nemerteans on the rough outline of Nemertean phylogeny, there are three major groups, three major clades in Nemerteans. One is called the Pylidiophora, because they are characterized by the presence of the Pylidium larva, which has this maximally indirect development and catastrophic metamorphosis. And then there is their sister clade, the hoplonemertea. Hoplonemerteans are the ones that are armed with stylets. Hoplite is a Greek soldier armed with a little dagger. So these are the dagger carriers. And then there's the Paleonemertea, which is the sister clade to Hoplo and Pylidiophorans. And the Paleonemertean larvae are similar in a way to Hoplonemerteans in that they are juvenile-like and they don't have catastrophic metamorphosis like the Pylidium. So traditionally, people referred to these larvae, the planuliform larvae, planula-like larvae, for their resemblance to Cnidarian planulae, as direct developers, and to the Pylidium larvae as indirect developers. But that is really, I really don't like using these terms anymore because I think it's misleading, and I'll try to show you why. Not only this is misleading in the ecological sense, because these planuliform larvae do spend a considerable amount of time in the plankton, and many of them do feed in the plankton, and they can disperse over large distances, connect and populate isolated populations. But they're also, it's also indirect development in the traditional embryological sense, that some of these larvae have go through a kind of a transformation that's maybe not as apparent as the catastrophic metamorphosis in the Pylidium, but nevertheless, there are certain larval structures that they have and lose over the course of their development. So as an undergraduate student, I wondered, how did the Pylidium larva evolve? Are there any kind of vestiges of pylidial body plan that can be found in the Hoplonemerteans or Paleonemerteans? At the time when I started this work as an undergraduate, people thought that people didn't know Pylidiophorans formed the clade. They thought some Paleonemerteans had the Pylidium larva and consequently imagined that pylidial development may be ancestral to the entire phylum. So with that in mind, I did my first research project looking at the development of Hoplonemertean, trying to find vestiges of the pylidial development. As I mentioned, the key features of pylidial development are these imaginal discs, which invaginate from the larval surface, and then they cinch away like little bubbles, and they fuse with each other. And the inner wall of these bubbles, or these imaginal discs, forms the juvenile body. That's the gut, black here. And then the outer wall of these bubbles, or imaginal discs, forms the amniotic membrane. That's the amniotic cavity, and that's the larval epidermis. And so that the presence of these invaginations, these invaginated rudiments of the adult, is one key feature. And the second key feature is the loss of the larval envelope, the larval epidermis during metamorphosis. So there was this larval body, and then it's lost and usually consumed by the emergent juvenile. So I looked at the development of the sister clade to what we now know is a sister clade of the Pylidiophora, and also the Paleonemerteans, to see if there's any vestiges of these rudiments and the loss of larval epidermis in other Nemerteans, to see, when and how Pylidium may have evolved. So this is a couple of illustrations from the first paper I published as an undergraduate student. This is the work I did at the White Sea Biological Station up in the Russian Arctic. This was in the mid-90s when the economic situation in Russia was pretty awful, and the university couldn't afford to pay the electric bill for remote facilities like the marine station up in the Arctic. So they cut off the electricity, and we studied with the sunlight, using mirrors, tilted mirrors on our microscopes. And we had a generator that ran a couple hours a day just for the kitchen to get started in the morning. And when we didn't have sunlight, we used candles and flashlights for our microscopes. And so this is a kind of a very low-key, low-tech study that I did. I looked at the development of this little intertidal Nemertean called Tetrastemma candidum. I collected them on the intertidal, brought them to the lab, and they laid these egg masses, mucus egg masses. And then I looked at the embryos through a very basic microscope using sunlight, candles, and so on. And this is what I saw, and I drew it with a pen and ink. So they have classical spiral cleavage. I'll talk a little bit more about what that means. But at the end of cleavage, you can see gastrulation. So that's formation of the primary gut. And then I saw some weird lobes there and some weird invaginations, which I didn't know what they were. And then surprisingly, after I thought the cells had become so small I couldn't see them, I noticed the cell outlines again. That was very strange to me, and I couldn't understand what that meant. And then the little juvenile developed and eventually hatched out and crawled away from the egg. And so I fixed these embryos and for histology and brought them back to Moscow with me and sectioned them physically on a microtome that was in our department as a trophy from the World War II, German microtome, really nice one. And it was really hard because these are tiny. They're like 200 micron in diameter, these eggs, and they're very yolky and they crumble. But nevertheless, I was able to see something. And what I saw on these sections was that, if you section an advanced embryo like that, you can see the proboscis and the sac in which it lies, the rhynchocyl. You can see the rudiment of the gut, these big cells full of yolk. And then you can see the epidermis. And then you can see these giant cells in the epidermis with pycnotic nuclei, which suggests that they're resorbing these cells and these nuclei. And so it turns out that, in fact, yes, at a certain stage in development, the larval surface contains these very large cells which are resorbed during what we called a hidden metamorphosis. And we wondered, well, could these giant cells represent a kind of a pileal envelope, a vestige of a pileal body, or perhaps a precursor of a pileal body? And ever since I saw that, I've always wanted to go back and look at development of more hoplonemerteans with better tools at hand. And so as a postdoc in Freddie Harbor Labs up in Washington in the United States, I finally got a chance. Hoplonemerteans are kind of hard to study because they don't like to spawn on cue. Nobody knows what the cue is. So sometimes you get lucky. And I got lucky one day. I collected these brown worms called Paranemertes peregrina, although now we know it's a cryptic species complex of something like at least 6 or 9 species, which maybe explains some of the difficulties I had with getting their cultures started. Anyways, but one day I got lucky. I went out on the mud flat and collected some, and they looked like they were ripe, and I put some in some females with what looked like big eggs in a tube and stuck it in my pocket, went back to the lab, and when I got to the lab and took the tube out of my pocket, there were eggs in it. And I thought, oh gosh, I gotta catch this. So I had a bunch of males and I chopped them up and got the sperm out and fertilized the eggs, and they developed. And I didn't realize at the time what luck that was, because I haven't been able to replicate that ever since. Not that I really tried very hard, but I'm kind of busy with other projects now. But anyways, so that one time I got these guys to spawn and develop normally. I was able to observe their development and document it with nice microscopes with DIC optics, and they developed into these lacetotrophic planariform larvae and settled and crawled on the bottom as juveniles after about 10 days. And so at the time, I had access to a confocal microscope, which is a really cool thing, which allows you to section your microscopic object optically instead of physically. So instead of a World War II microtome, I now had this beautiful tool where I could have a larva that was intact physically, and I could stain it with something fluorescent that glows in the dark. In this particular case, it's phalloidin again, which highlights cell boundaries because there's a lot of actin underneath every cell membrane. And so I stained these larvae with phalloidin and looked under the confocal microscope to see what I can see. And this is what I saw. This is a surface of a phalloidin labeled larva of Paranemertes peregrina at about 5 days of development. And what you see here is a bunch of big cells. So these big, what look like holes in the lace are the large cells. And then there's a whole bunch of little cells in between them, separating them. So it's kind of like big cells in the ocean of small cells. And that reminded me strongly of what I saw as an undergraduate student in this white sea species, Tetrastemma candidum, using those rather primitive tools. And this is a developmental series from the same species, Paranemertes peregrina, whichever one of those cryptic species it was. So early on, the larval surface apparently is covered almost uniformly with these fairly large cells. They're all uniformly ciliated, but you can't see the cilia in this prep because the cilia are not labeled with phalloidin. So you see the cell outlines. Apical organ is up, and this is the blastopore, which will become the mouth of the worm eventually. And as they develop, you can see that the large cells begin to be separated from each other by a large number of small cells, which progressively get more and more numerous, while the large cells progressively get smaller and smaller and are eventually entirely gone. So this is a 10-day-old larva in which you can see a bunch of muscles developed. Those are the strings there. And you can see that the surface is composed of a bunch of tiny little cells, and the big cells are gone. So it's basically what you're looking at is a kind of a larval epidermis composed of few large cells, which is being gradually replaced by a definitive epidermis composed of a bunch of small cells. So very similar to what I observed early on as an undergrad. And I've ever since looked at the development of many more species and dug through the ancient literature, and I discovered that pretty much every species of hoplonemerteans I look at, or anybody else had looked at in any kind of detail, apparently has this kind of larval epidermis that they either resorb gradually or shed. So this one species called Phylum californiensis, which lives on here in Oregon and along the west coast of North America, goes through a very brief stage at about 3 days of development, in which they just shed these chains of epithelial cells. And that is normal. That's not just because I squished the larva too hard or something. They all do that. And so they just basically get rid of their larval epidermis this way. This is a paper we published with Laurel Hibbert, who was a postdoc with Federico Brown. Laurel was a PhD student in my lab at the time when we did this work. Anyways, and this is just basically a list of hoplonemerteans which anybody had looked at in any kind of close enough at their development to see whether they have these transitory epidermal cells. And it turns out that pretty much everyone does. And so that seems to be the rule. And so when I discovered this about, I don't know, maybe some 12 years ago, and we published a paper about it, and I thought, well, gosh, I mean, is this the piledial homologue, the homologue of the piledial larval body and a kind of a hidden metamorphosis? Maybe, but there is more to the piledium than just the larval epidermis. Remember those imaginal discs that invaginate from the larval surface? Well, so it turns out that Planuliform larvae of Nemerteans, the hoplonemerteans, as well as Paranemertes, do have invaginated rudiments of some kind. Nobody had studied that in great detail to know what these rudiments are, what they give rise to. For some of them, we have some kind of guess, but for most of them, we don't, or didn't at the time. So this one is a Nemerteans californiensis, a west coast of North America species. This is about 3 days of development. These bright things here are the invaginations of the larval surface. They're bright because there's a lot of microvilli in those invaginated cells, and they're labeled with phalloidin. So that's the apical tuft here. That's the rudiment of the proboscis, a little invagination at the anterior end. That's the rudiment of the gut, actually. That's the lumen of the gut. There are these two mysterious anterior invaginations and two mysterious posterior invaginations. And this is a larva of a Paranemertean from Florida. And that's called Carcinoma tremophoros. And again, early on in development, that's 10 hours of development. It's warm in Florida, so things go faster. There's two invaginations at the anterior end, close to the apical plate. And nobody knows what they are for and what they give rise to. And that's the gut. So that's the blastopore at the posterior end. So it's kind of an advanced gastrula. So they all have these invaginations in various arrangements and numbers, and nobody knew what they do. And we wondered, well, could it be that these invaginations are somehow related to the invaginated rudiments in the piledial development? So Laurel Hebert, who was a PhD student in my lab at the time, she did this study where she cloned Hox genes from 2 species of Nemerteans, one with the piledial development called Maculaura alascensis, that's its larva. And she did in situ hybridizations with 6 different Hox genes, or maybe more even, I can't remember anymore. But there's 3 of them shown here. Maculaura alascensis labial, which is an anterior Hox gene, and then Maculaura alascensis sexcombs reduced, which is kind of a middle gene, Hox gene, middle of the cluster, and a posterior Hox gene called post2. And so these are slightly earlier developmental stages in the upper row, and the bottom row shows a more advanced developmental stage. The piledial body is really shriveled up, and that just is really difficult to do in situ hybridizations on them without losing that structure. But the juvenile rudiments, the gut here and the juvenile rudiments inside are retain their integrity more or less. So in all of these pictures, the apical organ is up. Right there, that cup there, and the gut, the larval gut, is to the right. Okay, so that's the stomach of the larva. And so the head of the juvenile is to the left, that's the anterior, and the trunk is to the right. And it kind of wraps around the gut, larval gut. And so when she looked at the expression of Hox genes in development of a piledian larva, she noted that all of the Hox genes that she cloned had no expression in the larval body, but they had expression in the trunk discs of the juvenile rudiment. So starting from early on in the trunk discs and the dorsal disc, these genes are expressed, and they continue to be expressed throughout development. And interestingly, they're expressed in a similar sort of staggered fashion as they are in other bilaterians, where they pattern the anterior-posterior body axis. So then she looked at the development of a planuliform larva of Nemerteans californiensis, which has those mysterious invaginations at the anterior and posterior end. And she discovered that Hox genes, she cloned, I think, 6 genes from this species, and discovered that they are expressed in vicinity of these posterior invaginations. And both the trunk discs of the piledium and the posterior invaginations, as we have found out, apparently give rise to the lateral nerve cords. which are the main part of the nervous system in the juvenile and adult body. So this suggests that perhaps these structures are in fact homologous. So there is a kind of similarity in this development of the planariform larva to the development of the pylidium, and that they both have invaginated rudiments of the trunk nervous system, which express Hox genes. She also stumbled upon a similar expression of the so-called 636 gene, which is expressed in anterior invaginations of the hoplonemertean and the cephalic discs of the pylidium. These genes are often expressed in anterior sensory structures in other kinds of animals and other kinds of invertebrates, alophotrochozoans in particular. So that kind of shows that there may be some kind of correspondence, at least functional, between the anterior invaginations of a planariform larva and the cephalic discs, the anterior most imaginal discs in the pylidium. So we published a paper kind of summarizing these findings, and so in color here you can see that we shaded the anterior invaginations in purple and the juvenile head inside the pylidium as purple, and the posterior invaginations as in cyan, and also the trunk of the juvenile inside the pylidium in cyan, suggesting that perhaps somewhere before the divergence between hoplonemerteans and pylidiophorans, the common ancestor of these groups had the kind of development, whether it was pylidial or not, but they had development in which they had these tucked away invaginated rudiments, which gave rise to the head structures and the trunk structures. And that's maybe the common origin of the imaginal discs in a way. So that kind of summarizes our insights of how the pylidial body may have originated, but there is more to it. There is one other vignette that I would like to talk about. And I don't know how we're doing on time. Maybe I should check. Okay, how we're doing on time, Joanna? Do we have an, okay, all right. So yeah, so I'll talk a little bit more about other approaches to studying how the pylidial larval body may have originated. And that has to do with early embryonic development, spiralian cleavage. So, and the evolution of cell fate in the pylidiophorm Nemerteans. So Nemerteans are spiralians, as I mentioned, and spiralians as a group are characterized by this very conserved pattern of early development, embryonic development, in which there's a characteristic cleavage pattern called spiral cleavage, where the egg divides first symmetrically or sometimes asymmetrically into two cells. These pictures here represent so-called equal spiral cleavage, in which the first two blastomeres are equal in size. Then they divide into 4 blastomeres. And then once you have these 4 blastomeres, they act as progenitors of lineages and sort of more or less synchronously bud off cells toward the animal pole of the egg. And these quartets, so that's these dark shaded cells here. The first quartet, called micromeres, because they're smaller than their sister cells, the macromeres, which are at the vegetal pole. And then later on, the macromeres, these first 4 blastomeres, divide again to produce a second quartet of micromeres, and then a third quartet of micromeres, and then a 4th quartet. And then things become asynchronous and kind of lose that spiral arrangement. The spiral arrangement originates from the twist of the cleavage spindles during these cell divisions, in which the daughter cells, the animal cells, the micromeres, are twisted first to the right from their respective sister cells, kind of clockwise, and then in this next division counterclockwise, and then again clockwise. So they alternate these clockwise and counterclockwise cell divisions, And the cells are kind of arranged in a certain spiral-like mosaic in the embryo. That's where the name spiralia comes from. It's from this cleavage pattern. Anyways, but it's not just the cleavage pattern that's conserved, remarkably conserved across a wide array of animal phyla with widely divergent adult and larval body plans. It's also the cell fates of these cells. So you can identify, the beauty of spiral cleavage is that you can identify homologous cells in the development of different phyla, say annelids and Nemerteans. And you can ask a question, what does the cell become in the larva or the juvenile? What does it give rise to? What is its fate? And you can label individual cells in the embryo. There are tools to do that, say with intracellular injections of dyes or some other way. And you can, if you can do this successfully, you can trace the development of these labeled embryos and identify the fate of these cells. And so one of the most conserved fates in the spiraline development are the cells that give rise to the so-called prototroch. So many spiraline larvae are characterized by having a transverse ciliary band anterior to the mouth called the prototroch. So this is kind of a hypothetical idealized trochophore, which has a prototroch, a ciliary band anterior to the mouth, and another ciliary band posterior to the mouth called metatroch. Then there's a kind of a foot groove that's ciliated, and a ventral neurotroch, and then the posterior telotroch. But I want you to pay attention to this prototroch here right now. This is a chitin trochophore larva, so a mollusk. It also has a prototroch, this transverse ciliary band composed of very large multiciliated cells. And the prototroch is important in the larval life because not only it's its primary swimming organ of the larva, but in larvae that require food to develop to metamorphosis, the prototroch is often involved in feeding. Here's another annelid larva with a prototroch, which is also anterior to the mouth, as always. Pretrochal, there's a telotroch, a posterior ciliary band here. Here's another annelid larva, Polygordius, and that's kind of a view from the animal pole. But this ciliary band here all around is the prototroch. And so the presence of the trochophore larva with its, at least the prototroch, is often thought to be a synapomorphy of the spiralia, one of the few synapomorphies aside from the spiral cleavage itself. And the prototroch develops in pretty much every spiralian that people have looked at, develops from this one cell lineage called the primary trochoblasts. So at the 16 cell stage in the embryo, these four shaded cells called one little Q2 in the spiral cleavage nomenclature, they are the primary trochoblasts, and they have specific fate. What they do is they divide once, making 2 cells each, and then another time. So there's 4 blocks of 4 cells, and then these cells become ciliated, multiciliated. They grow cilia. So you have this little, not even a gastrula yet, that already has a bunch of cells roughly in the equatorial position with cilia. And then these cells rearrange themselves in a kind of a circle to form the core, the central part of the prototroch of the trochophore larva. So this is a molluscan trochophore. This is a limpet. And these shaded cells here are the primary trochoblasts that contributed to the core of the prototroch. There are some cells up at the animal of the prototroch and vegetal of the prototroch that contribute as well. There's so-called accessory trochoblasts and secondary trochoblasts. But the core, the main cells are usually the 16 cells derived from these 4 primary trochoblasts. And so one thing I did as a graduate student was spend a few months at the Smithsonian Marine Station in Fort Pierce, Florida, where I got a chance to study the development of a Paleonemertean. So this was at the time when I've just kind of discovered these large cells in the development of hoplonemerteans on the surface. And I was wondering whether some kind of transitory epidermis may also be present in a Paranemertean. And there are so few studies on Paranemertean development. And one of them mentioned some kind of transitory epidermis, but it was, a very old study, just like hand-drawn images, pictures of something that wasn't entirely clear to me what it was. So I went to Florida and collected this Paranemertean that's common there on the sand flats, Carinoma tremophorus, got them to spawn, got them to develop in the lab, and then in collaboration with Mark Martindale and my PhD advisor, John Nuremberg, we labeled these cells, Mark did the injections. He's a famous embryologist and had done cell lineage studies on all kinds of marine invertebrates. So he came down to work with me and he injected a fluorescent dye into the primary trochoblasts in Carinoma, in this Nemertean. And I raised these larvae and then stained them with phalloidin and looked at them under the confocal microscope to see what the primary trochoblasts give rise to. And what I saw was really astonishing. What I saw is that each of these primary trochoblasts apparently gave rise to a block of 4 large cells kind of in the equatorial region of the larva. And together, without any kind of dye injected, if you just label a larva with phalloidin and look at the cell outlines, I was astonished to discover that the larva had a kind of a ring. It's oblique with respect to the anterior posterior axis of the larva, but it's anterior to the mouth, which is right about here. And it's composed of 40 large cells. And you can't see that at all at the scanning electron microscopy level or light microscopy level. So the whole thing is uniformly ciliated. So the presence of these large cells is obscured by the uniform ciliation. But if you label them with phalloidin and look on the confocal microscope, you can see them. So basically, what we see here looks almost like a prototroch, a hidden type of prototroch in a Nemertean. And nobody had ever seen a prototroch in a Nemertean larva before that. So I was looking for a transitory epidermis. What I found was a kind of a hidden prototroch derived from the trochoblast cells in the spiralian embryo, which was remarkable because it kind of connected for the first time Nemertean larvae to the larvae of other spiralians in a way that had just not been there before. It kind of revealed a deeper homology, and was one more piece of evidence to suggest that Nemerteans are closely related to perhaps animals like annelids and mollusks, which have these trochoblasts and prototrochs, and not so much flatworms that don't seem to have a prototroch. Anyways, but the thing is, then we wanted to know, well, but what about the Pylidium? So maybe Paranemerteans have retained this kind of ancestral hidden trochophore type larva with these trochoblasts. But what about the Pylidium? Pylidium has this prominent ciliary band that spans the lobes and lappets of the larva, but those cells are composed of, that ciliary band is composed of many very small cells. And that doesn't seem like trochoblast type of thing at all, because trochoblasts become cleavage arrested very quickly, so they stay large and become multiciliated. And it doesn't seem like that could possibly be the case in the Pylidium. So my husband and I, George Van Dasso, did a study on the cell lineage of a Pylidium with a specific purpose to see what happens to the trochoblasts in the Pylidium. So we got a local species, Maculara alaskensis, and we can culture these things in the lab easily. We can inject their eggs with all kinds of fluorescent markers and raise these labeled embryos and then look at them under the confocal microscope. And that's what we did. So we took the embryos and injected them with a combination of markers. This happens to be M-cherry, MCAC protein, fluorescent protein, which kind of is in every cell, and we injected that into the zygote, and and then it had a mixture of photoactivatable GFP protein as well. So that was injected into a zygote before this any cell divisions, and then we allowed these embryos to divide, and until they had reached the 16 cell stage, and then we used a laser to activate fluorescence. So if you hit this photoactivatable dye with a laser, it becomes fluorescent and glows green. It's green fluorescent protein with a photoactivatable domain. So we activated fluorescence in the trochoblasts and then allowed these embryos to divide and develop further, and then imaged them. So this one is an 8-hour-old embryo in which the primary trochoblast, one of the primary trochoblasts, had been labeled in such a way. And you can see here the progeny of that primary trochoblast. And so already at this stage, we could see that primary trochoblasts in the Pylidium larva do something different from what they do in most studied spiralians. They don't just stop there after 2 cell divisions. They keep on going and dividing. So there is already 8 cells here derived from a single primary trochoblast. And it turns out they just keep going and they keep dividing. So they do not undergo this cleavage arrest that the trochoblasts in other animals do. And indeed, these trochoblasts contribute to the primary ciliary band of the Pylidium larva. But instead of contributing just the 16 big cells, they contribute dozens of small cells to the ciliary band. And what was interesting is that not only do they give rise to the ciliary band, but they also give rise to these kind of indentations between the nascent lobes and lappets of the larva. These indentations we called axils, from the Latin for armpit, in reference to the axillary growth zones in plants. Between the stem and the leaf, they have this axillary meristem and armpit meristem, where there's a bunch of dividing cells. And so we named these regions axils and noticed that primary trophoblasts were contributing to these armpits. And axils in the Pylidium, as we have discovered, are the pylidial growth zones. So this is a Pylidium larva that was labeled with a dye called bromdeoxyuridine, which intercalates into DNA of actively dividing cells where DNA is being replicated. And then you can let the cells divide and all the progeny receive these intercalated modified nucleotides, and then you can label them with an antibody against them and highlight the progeny of the dividing cells. And so if you do that, you can basically identify nests of actively proliferating cells. So when we did this was just a student project and kind of an accidental discovery, but we've discovered accidentally that these axils, these armpits or lappet pits in the Pylidium contain the majority of the dividing cells. There are some around the apical organ of the Pylidium and some in the stomach and the main ciliary band and scattered in various other parts, but the main regions of the dividing cells are concentrated in these axils. And it turns out that you can actually see these axils and these putative stem cells of the Pylidium on the scanning electron micrograph. So this is a Pylidium larva viewed through a scanning electron microscope. And if you zoom in to these regions here, these axillary regions, you can see that most of the cells, so these are the ciliary bands, and that's the cilia of the epidermal cells of the Pylidium, are multiciliated. They have multiple cilia each, and such cells in animal kingdom cannot divide. Once they have multiple cilia, that means they're cleavage arrested, because during cell division, the cell uses centrioles to create a spindle and segregate its chromosomes into daughter cells. And each cilium has a basal body, which is basically like a little centriole. And so if you have multiple cilia, you have these multiple basal bodies underneath cell membrane, which could each act as a microtubule organizing center to create a spindle and mess up the cell division. So once an animal cell has multiple cilia, it cannot divide. So if you see multiciliated cell, you know it's not going to divide again. And we've discovered this accidentally by sort of wondering, well, how does the Pylidium larva grow? How can it grow if all of its cells are multiciliated? But it turns out not all of the cells are multiciliated. Some are just have a single tiny little stubby rudimentary cilium. And these cells are located in these axillary regions, and you can label them with, and their progeny with BRDU. So back to the trochoblast story. Basically, the trochoblasts give rise to lots of cells, not just 4 each, like another spirines, and they contribute to the primary ciliary band and the axils, which are the pylidial growth zones. So that's a kind of a fundamental difference in the behavior of one of the most conservative cell lineages in the spiralian development that leads to the creation of this unique body plan, the pylidial body plan. And what's more is that these axils, the cells from these axils populate the imaginal discs as well. So the imaginal discs, if you recall from earlier in this talk, are the juvenile rudiments. So the juvenile rudiments invaginate from these axillary zones and contain the progeny of presumably the primary trochoblasts. So the primary trochoblasts are not strictly larval as they are in other animals. In mollusks and annelids and in Carinoma, the primary trochoblasts, they have this single job in which they go through one or two cleavages, make lots of cilia, form the primary ciliary band of the larval body, and then they disappear during metamorphosis, contributing not at all to the juvenile or adult. But in the pylidium, that's not at all what they do. In the pylidium, they contribute to the continuously growing larval body by contributing to these axillary growth zones, and they also contribute to the juvenile body. And so, so we think that this change in the cell fate of this one of the most conservative lineages in spiralians, the primary trochoblasts, may have led or is associated with the evolution of this novel larval body plan, the pylidium. This continuously growing planktonic larva that has these distinct juvenile rudiments that form the juvenile body. So that's kind of the insights we got on how the pylidium may have evolved. We talked about the transitory epidermis that may be homologous to the pylidial larval body, and the invaginated rudiments found in hoplonemerteans and Paranemertes that also may have been present in the most recent common ancestor of Nemerteans. And I talked also about the change in the behavior of one of the most conservative cell lineages, the primary trochoblasts, that may have led to the evolution of this novel larval body plan, the pylidium. But there is one more vignette, and I don't know if we have time for that. Joanna, how are we doing on time?
It's up to you, because like we are, most of what we are thinking was like 1 hour and time afterwards for discussion. But then it's up to you, Sveta. It's really interesting. So
okay, right. So this is probably maybe another 15 minutes worth of. So if you guys want to, I'll tell a somewhat different story about, so far I've been talking about how do you make a pylidium from something like a trochophore. And this is a story of how do you make something that looks like a trochophore from a pylidium. So I don't know if you guys read that paper I sent, from trochophore to pylidium and back again. So this is the back again part. So again, this was an accidental discovery that we made here in Oregon. As we often get depressed in the winter, it's cold and rainy, and the teaching gets us down and the kids are crazy. And so sometimes we just go and look at plankton for fun. The plankton is really great here in the winter because there is no diatoms. They finally die off. And so the nets are not clogged anymore, and we can find all kinds of really cool larvae. And so it's just kind of fun to sample plankton and look at it and just see what we can find. So one day, many years ago now, George and I were in the lab looking at plankton. And each of us found this interesting kind of trochophore-like thing. I mean, it had cilia all over. It was swimming like a trochophore, had an apical tuft, and had two transverse ciliary bands, kind of like some annelid trochophores have. One more or less equatorial, kind of like a prototroch, and one at the posterior end, kind of like a telotroch. And we each noticed it independently in our bowls and set it aside thinking, oh, that's a funny looking trochophore. And then we compared our trochophores under the microscope and noticed something weird. We noticed that there was a little worm, what looked like a little worm inside the trochophore. The trochophore also had these very, very strange behaviors, like it singed it stopped its ciliary beat every once in a while and then singed a band of muscles along its equatorial region and, underlying the prototroch-like belt and the telotroch-like thing, and also sometimes pulled in its apical organ, kind of the way a pylidium larva does. And so we thought, oh my God, is this a kind of a licitotrophic pylidium? And this second video here shows what happened next. As we looked at this larva and tapped on the slide on the cover slip, we prompted, in fact, metamorphosis. And indeed, what happens here is that a little worm that was seen here inside is coming out of the larval body. And as it's emerging, posterior end first, it's simultaneously swallowing its larval body, just like a typical pylidium would. But it looks nothing like a pylidium at all. It looks like a trochophore larva. So how can this be? So licitotrophic development has been noted in a few species of Pylidiophorans before, but none of them were known to have two transverse ciliary bands. So we were the first ones to discover this type of larva that is basically a kind of Pylidiophorans, which we guessed from the type of metamorphosis it had. So you can see the remaining part of the larval body disappearing into the mouth of this young Nemertean worm that had just emerged from its larval body. So this is the first time somebody discovered such a trochophore-like larva among the Pylidiophorans. And we had only two larvae, and one of them metamorphosed. And I barcoded that juvenile and to try to see if I can identify which species it belongs to. And remarkably, I got a match, species level match to a local undescribed species from Oregon, which we refer to as Micrura sp dark, a very boring brown worm, of which I've only ever found one at the time. And it was sitting on my sea table for a long time, and I didn't know what to do with it because it was just so boring, and it was just one, and what do you do with such a thing anyways? But I eventually chopped off a little piece of it and saved it for DNA barcoding just so that I have a representative of it. And now I know why I did that, because when we found this weird larva, we were able to match it to this particular species. And then we said, aha, so yes, it is definitely a Pylidiophorans. It's a local one. We know where it lives. Oh, so we thought. It's this one little cove, and we found it once, and we kept going back there trying to find more of them so we could get them to spawn in the lab and find more of these larvae. And it took several years until we discovered a local population in a neighboring cove. And it took a dedicated master's student who went out there in the rain and climbing down a mud trail, very treacherous mud trail in the winter, and dug in amongst the roots of the surf grass at low tide to find more of these brown worms and bring them to the lab and spawn them. And she got them to develop in the lab, and we were able to follow its development. And I'll show some pictures, but this is the first confocal microscope picture of this larva. So we had two initially, one I DNA barcoded to find out what it might be, which species it might belong to, and the other one I preserved for confocal and labeled with phalloidin, and this is what it looks like. And it's a very interesting trochophore-like thing. It has these two transverse ciliary bands, but unlike a typical trochophore, say a molluscan a limpet larva, in which the prototroch is composed of very large and very few cells, the prototroch, and this is in quote marks on purpose, the prototroch, the equatorial ciliary band, and this unusual larva we discovered is composed of lots of very small cells. And at the posterior end, there is the so-called telotroch, which is also composed of very many, very small cells, smaller than the cells elsewhere in the epidermis. So that's kind of the opposite of what real trochophore looks like. And also, we discovered that the blastopore in these unusual larvae is posterior to the telotroch, whereas in the typical trochophore, it is posterior to the prototroch. So it looks like basically a convergent type of trochophore here. And when I got this master's student who was interested in trying to figure out how this trochophore comes about developmentally, and she was successful, she spawned many of these worms in the lab and got them to develop, and we were able to preserve many embryos at various developmental stages and stain them with phalloidin and confocal them to look inside. What we discovered was that these larvae, go back. These larvae, I just want to pause this video here, go through a very brief stage in development in which they resemble a chubby little pylidium with lobes and lappets and an apical organ and a gut. So this is a kind of a fly-through movie here, from the surface through sectioning through optically through the to the other side of the larva. Okay, and that's a side view. So there's the gut inside, just like in the pylidium, and then there's a bunch of what looks like imaginal discs, the cephalic discs and the trunk discs, and that's the entrance into the gut here. So this is a slab confocal projection with a bunch of sections stacked up that shows the gut, the apical plate, the anterior and posterior lobe, and the lappet, with the small cells of that kind of lining up the margin of this lappet. And this is a three-dimensional reconstruction of this confocal stack, a different confocal stack of this from the species. And you can really see how this looks like a chubby little pylidium. It has anterior lobe, posterior lobe, and two lappets spanned by these small cells, which will create the ciliary band eventually. So it looks like almost a continuous ciliary band of a typical pylidium, except this thing is non-transparent, full of yolk, and fairly large. So these are about 300 micron eggs, as compared to something like 100 micron or smaller egg. And they reach the stage in which they have all the imaginal discs in about 3 days of development, whereas a typical planktotrophic pylidium takes about 3 weeks to reach that stage. So in fact, this is very much like a pylidium, but very briefly, and it's a non-feeding pylidium. So what happens next in the development is within just a few days, this pylidium-like thing transforms into this trochophore-like thing. So the two portions of the ciliary band that spans the anterior and posterior lobe of this chubby little pylidium fuse together laterally to create this equatorial ciliary band, the so-called prototroch in quote marks. And the portions of the ciliary band which span the lateral lappets of this chubby little pylidium, fuse together to make the posterior ciliary band, the so-called telotroch, the in quote marks. And that takes only a couple of days, literally. So you really have to catch that stage to see that transformation happen. So this is kind of a summary diagram of how this Pylidiophran basically got to have a trochophore-like larva convergently. So this is a typical planktotrophic pylidium, which swims around for several weeks or several months in the plankton, feeds, and develops gradually these juvenile rudiments, which fuse to form the juvenile body. And this is the so-called licitotrophic pylidium, which doesn't require food because it relies on the yolk inside the egg. And it goes through development of the juvenile very rapidly while it swims in the plankton. The juvenile, just like in a typical pylidium, develops from the same number and arrangement of imaginal discs, but the ciliary band, which they don't need for feeding, is transformed into two transverse ciliary bands, which apparently is a lot more advantageous for swimming compared to the single convoluted ciliary band that the pylidium larva uses for feeding. So that makes sense, and it's very similar to the evolution of non-feeding larvae in other phyla of marine invertebrates. There are parallels to this, especially in echinoderms, where the many sea cucumbers have non-feeding licitotrophic larvae, which have these transverse ciliary bands, whereas their other species have a feeding larva with a long convoluted ciliary band. And in development of the feeding planktotrophic sea cucumbers, you can see in the development of a single species how the single convoluted ciliary band of the auricularia larva, a feeding auricularia larva, transforms in development into several transverse ciliary bands of the non-feeding stage called doliolaria. So that's kind of an analogous transformation here. So that highlights convergent evolution of a trochophore-like larva in the Pylidiophorans. This is the last slide, and it's just, this is a phylogeny of Pylidiophorans. This is a work by another PhD student of mine, Tara Hibbert, and she was interested in figuring out how many times licitotrophic development may have evolved within the Pylidiophora. When we began this work, 20 years ago, only 3 species of Pylidiophorans were known to have non-feeding or licitotrophic development. But at this point, we know of over 20 species of various Pylidiophorans with non-feeding development. And many of these we discovered ourselves here locally in Oregon and in the Caribbean. And some were discovered by colleagues around the world. And so we can barcode these larvae, even if we just have one out of the plankton and don't know which species they belong to, and then we can place them on the phylogeny and try to optimize the character evolution to see how many times this non-feeding development may have evolved from the typical classic planktotrophic larva. And however you optimize the character evolution, it turns out that there is at least 4 to 9 times that licitotrophy may have evolved within the Pylidiophora. So, and every time what happens is that they lose this hat-like shape and they become kind of bullet-like and compact, and they either evolve uniform ciliation or they evolve various number of transverse ciliary bands, sometimes 1, sometimes 2. And we, over the years, like I said, we discovered many of these novel non-feeding larval types, and it looks like it just happens time and again within the Pylidiophora, as is the case with echinoderm development as well. And that's all I have to tell you guys. And I just wanted to thank the members of my lab. Most of these are former members at this point. Laurel Hibbert, my first PhD student who did the work on Hox genes, and April Bird, who was a master's student and studied with us how the Pylidium larva grows and discovered those axils. And Marie Hunt, who was a master's student who studied the development of this unusual licitotrophic Pylidiophran with a trochophore-like larva and Tara Hibbert, who did a lot of DNA barcoding of local Nemerteans in Oregon and tried to match larvae to adults and had studied evolution of licitotrophy in the pylediophorans. And above all, my husband, George, who is a professor here at OMB and a lab mate of mine, and who has been involved in much of this work over the years, even though he's not a Nemertean person. He's really a cell biologist, but he's just very curious about everything and keeps getting involved, and I can't help it. So I'll end here, and thank you guys for your attention. I know it went probably way too long, but I'm happy to take any questions you may have.
Thank you very much, Sveta, for the wonderful talk. It's very nice to hear all this, and very nice how to hear how things, small information build up to answer a bigger question. So thank you very much. I got lost here. I had everything set and then I lost it. So thank you very much also to all that have been watching the talk with us. I hope you enjoyed as much as we did. Muito obrigada a todos que estavam por aqui assistindo com a gente, a palestra da Svetlana. Espero que todos tenham gostado tanto quanto a gente. Pronto.